Welcome to our JoVE webinar, everyone. Thank you for attending. So today we're going to have Randal Halfmann talking about mapping complex protein phase behavior in vivo. So Dr. Halfmann published with JoVE, did a video publication with us in 2019. And today he's going to be talking about recent updates and modifications to the protocol that have happened over the last 4 years. So we will be recording the webinar, and we'll send out an email with the recording afterwards for your records. And also after the talk here, there'll be a brief Q&A session. So feel free to add your questions in there, and we will address those at the end. Okay, well, thanks again, and welcome to the webinar, and take it away, Randal.
All right, thank you, Ronald. Thanks for the introduction. I am eager to share with you guys some updates to the protocol, the procedure that we published with JoVE in 2019. So using DAMFRET to map complex protein phase behavior in vivo. Of course, I'm Randal Halfmann. I'm an associate professor at the Stowers Institute. And I'm also asking my postdoc, Shriram Venkatesan, to be here as well. So Shriram was one of the key developers of the original DAMFRET protocol, and he was the first author on this JoVE article. And so what I'll ask is that if you have any questions throughout, you can either hold them until the end, and then we can talk about them in the Q&A, or if you want to know right away, you can feel free to have your question in the chat, and I'll ask Shriram to answer it to the best of his ability in real time. So thanks, everyone. So I want to introduce first, why do we need this approach? How do we use DAMFRET to characterize phase behavior of proteins in cells? What is it? How does it work? And in addition, a few little updates that we've had in the last few years. And I also want to tell you about some of the really fun applications that we've found for DAMFRET, specifically as it was initially intended, that we can use it to distinguish kinetic from thermodynamic barriers of phase behavior, of phase separation. And so I'll tell you a little bit what I mean by that. We can use it to see condensates that you can't observe by microscopy, which we're actually finding are surprisingly common. So these are an emulsified phase. You can use it, of course, to identify sequence determinants of phase behavior, as well as biological, cellular factors that may be regulating phase behavior. And then most recently, we've found that we can actually map proteotoxicity to specific states of a protein of interest, which is quite powerful in terms of understanding how phase separation can contribute to proteopathic diseases. And so what is complex phase behavior? I think anybody who's here is familiar with condensation and phase separation in a broad sense. This illustration is from a recent review showing that there's really two pathways to form a condensed state of proteins, a condensed multimeric state of proteins. So most of you are undoubtedly familiar with condensation, so liquid-liquid phase separation. And that really corresponds to a transition from a soluble state, most often a monomer or a discrete oligomer, to some condensed state of really infinite stoichiometry. So a liquid droplet is how this typically manifests in cells or in vitro. And importantly, there's no ordering transition here. This is purely a transition in the density of molecules of that polypeptide. Now, you can also have an ordering transition where proteins, either in a soluble state or in a liquid state, can become highly ordered. So they can acquire crystalline order in 1, 2, or 3 dimensions. And that will most often manifest as an amyloid. And that's really the major interest in my lab, is understanding that transition. Now, of course, this transition can be extremely challenging for proteins to undergo because it's simultaneously a transition in density and ordering. So there's both an intermolecular and intramolecular entropic component of that transition that makes it very challenging in a kinetic sense. And so what happens is proteins will often get stuck in these intermediate states. So whether it's from some sort of monomer to oligomer, and then the oligomers can kind of sort of slowly try, if you will, to acquire order, or liquid droplets can go through some sort of aging process, some sort of maturation where there's incremental acquisition of order within it, leading ultimately to something that's more and more like amyloid. And if you think about this from a thermodynamic sense, that is to say on a phase boundary, and when we think about just liquid-liquid phase separation, there's only two regions of this phase boundary. So there's a monomer, and then there's a condensed state, the condensate. And there's some critical concentration or critical temperature, in this case, in both cases, denoted by this blue line, the phase boundary. Now, for lots of proteins, and this is where the complexity arises, is that there's often one or maybe even many ordered states, and those tend to have much greater stability, right? So you're ordering, you're acquiring much greater avidity of interactions within between the polypeptides. And so as a consequence, the phase boundary for that transition, or for that state rather, is much lower. So they tend to be more stable. And now, and again, as I said, because that transition can be difficult to happen, that is to say that it has a large nucleation barrier, proteins can bypass that in the cell, so they can become what's called supersaturated, and then enter this condensed state. And then, of course, there's some maturation back to this amyloid form. And so it's that process, that this competition between different phases in which a protein, under conditions where a protein can enter either of those phases, that results in the complexity that a lot of interesting biology comes from and a lot of pathology comes from. So that's really the topic of today's discussion, is how can we begin to dissect that complexity? And so just to illustrate why we care about this. So, one of the end states of these kind of phase transitions is amyloid. And this is, of course, very closely associated with pathology. Over 30 human diseases are driven by amyloid formation, or at least associated with amyloid formation. And what's become clear, thanks to lots of breakthroughs in structural biology, particularly cryo-EM, is that different amyloids from patients, even of the same protein, can have very different structures, right? So it seems to be similar conditions, physiological conditions, and the same sequence, and yet there's a different structure of the amyloid, and that structure is associated with a specific phenotype, a specific pathology of the protein. And because amyloids are self-templating, right? So once this ordering happens, there's a transition where other polypeptides will join that ordered state. The structure that initiates initially influences what will happen over time, right? And so that is to say that maybe the most important step of aggregation, proteopathic aggregation, is the nucleation event. How does that conformation first begin, right? So how does it start? And then ultimately, right, the holy grail is to understand how can we prevent it from starting, or how can we influence that pathway. And now, why is this so hard? Why do we need a new assay to study this phenomenon? It turns out that understanding nucleation is extremely difficult. You can imagine that structural biology can tell us what the rate limiting conformational transition is towards amyloid, but that's not actually true. What you see is the end state, right? Once the protein has achieved a sort of a steady state or equilibrium, you can understand the structure, but it's really hard to know what the rate limiting structure, what were the rate limiting conformational fluctuations that allowed that to happen. It's not necessarily the same thing that you see in the end. Protein biochemistry is very challenging to do this because in a traditional in vitro system, like in a test tube, you're working with volumes that are more than 1 billion-fold larger than the volume of a cell. And importantly, because this is a kinetic barrier that limits these things, you've basically eliminated the kinetic barrier to nucleation when you work in larger than cell-sized volumes. And you can imagine that you can explore this computation, which is of course true, this various coarse grain and umbrella sampling approaches. But to date, no one has ever been able to see spontaneous nucleation happen in a full-sized protein molecule undergoing nucleation to amyloid. Right? So it's just too many degrees of freedom. It's far too expensive computationally to understand this at the at a very high level. So that leads us to needing to develop a new approach. And so what we've done is develop DAMFRET to try to make this easier. And so what DAMFRET does is it gives us, it allows us to probe protein phase behavior in cells, so in sufficiently small volumes under the exact physiological conditions that are, or at least most of the physiological conditions that are important for phase separation and amyloid formation. And it also gives us a direct readout. So what that means is that we can now look directly at aggregation without there being any sort of intermediary, right? And it's in a simple, it's a simple readout so that we can actually do this across biological systems and use the same assay for the same protein in human cells, yeast cells, bacterial cells, what have you, right? So that's very important. Okay. And so to develop this tool, we took inspiration from movies like this. This is a movie of actually one of the first functional human prion proteins, and I'm using prion very loosely in this case. It's actually a protein in innate immunity that is required for inflammation in response to things like viruses, and it functions by forming a prion-like state, so a self-templating, self-activating state. And here you can see this protein undergoing that transition in yeast cells. So initially it's expressed to high concentrations. You can see it's diffuse, and then spontaneously it forms this punctum. And that punctum coincides with an ordering and condensing transition. And so you can imagine trying to extract features of that pathway, right, from monomer to ordered by following individual cells over time as they increase concentration, and then there's going to be some spontaneous or semi-stochastic transition. And you would see something like this for different cells. And you can see that there's some stochasticity about that transition point, and that's going to be important, as I'll describe in just a minute. Or rather than taking thousands of movies, you could just take a snapshot of a culture undergoing this transition using DAMFRET. And what you see is now for hundreds of thousands of cells, or tens to hundreds of thousands of cells, where in this transition they are at that moment in time as a function of the concentration of protein in the cells. And so what you see in this case is two states. So every point here is a cell. And within each cell, you can see what is the concentration of the protein in that cell. And you can see the extent of assembly of the protein. So that's on the y-axis. And you can appreciate that there's really two populations of cells here corresponding to the soluble state and the punctate state. And what you can appreciate is that there's a gap, right? So these two populations are discontinuous. And what that tells us is that this transition is nucleation limited, right? So there's some really difficult, kinetically difficult event that has to happen for nucleation for the first little bit of that crystalline polymer to form. But once it happens, the other proteins join on extremely rapidly. And so you don't see any protein or any cells hanging out in the middle between these two populations. Once nucleation happens, they jump from the bottom population to the top population almost instantaneously. And you can also appreciate that there's some overlap between these two populations, and that's important. So what that tells us is that that nucleation event, or that phase transition, is not limited, in this case, purely by the concentration of the protein. Instead, there's some kinetic barrier resulting from intramolecular conformational changes that are rate limiting, right? And so the magnitude of that overlap actually tells us something about the magnitude of the conformational changes, right, that are rate limiting for nucleation. So that's how we can kind of begin to dissect what's happening at the structural level during these transitions. And so there's some nuance there that if I had lots of time, I would tell you a lot more about, but feel free to ask me or Shriram in the questions. And so basically how this works, so DANFRET stands for distributed amphifluoric FRET, and the distributed refers to the x-axis. So we're probing a wide range of concentrations. And amphifluoric refers to the particular way that we're, the technique we developed to assess self-assembly. And so basically what we're doing is we're expressing in this diagram, in yeast cells, our protein of interest fused to a photoconvertible protein, MEOS 3.1. And this protein has a unique property that it starts out green, but if you hit it with violet light, it will transition to a red form, irreversibly to a red form. And that transition is probabilistic for every molecule. So what you can do is within a culture of cells, you can just illuminate that culture for an empirically optimized amount of time to transition a fraction of the molecules from green to red. And then you have both a FRET donor and a FRET acceptor expressed by the same genetic construct, by a single gene, right? And that's really nice. So because of that, we can then use a system that, in this case, a noisy high copy number plasmid, such that we explore roughly 100 to 1,000-fold range of concentration between cells. But you can also just do this by transfection into like HEC293 cells. You grow these cultures up. In this case, we arrest cell division upon protein induction, such that every cell can be treated as an independent reaction, independent experiment. And then we subject them to limiting photoconversion, and then we analyze those cells by flow cytometry, where we collect both the emission of the red state, of the green state, and of the red state when excited by green, right? So FRET. And we can show that, because this is a single genetic construct, we have exactly the same ratio of donor and acceptor in all cells, regardless of concentration. And that's what I'm showing here in this panel E. And we can also show that the state of the protein does not influence photoconversion. So these are multiple different proteins ranging from monomer to amyloid. There's even some condensate in here. And so you can see that they all basically fall in a line. So the state of the protein doesn't change this, which means we can really use this to probe the intrinsic properties of the protein. So briefly, how we actually analyze the data after collecting it on flow cytometry. If this were imaging flow cytometry, you're going to start by looking at just the cells that are in focus. You're going to take just the circular, so the spherical events, and that's this panel C. And importantly, that's eliminating debris and also clumps of cells. You don't want to analyze multiple cells simultaneously, so we're getting single cells. We're looking at just the live population, so we're using autofluorescence in this case to gate out the dead cells or dying cells. And then within that live single cell population, you can actually see what's happening at the protein level. So here we're looking at donor intensity versus acceptor intensity, or as a function of acceptor intensity. And you can see there's these two populations. So there's this linear increase, but then there's a regime for this protein where donor intensity drops precipitously. So that's as a consequence of FRET. So the donor, the excited state of the donor is now radiating that energy to the acceptor. And so then you have and consequently, and also an increase in the acceptor. And then you can plot this, replot that data in a way that makes sense biochemically. So you can plot it as a function of concentrations or just the acceptor itself, as well as the ratiometric FRET. So this is now FRET divided by acceptor. And so that's kind of the plot that I just showed you, where you have these two states. And now you can make sense of how this protein undergoes phase separation as a function of concentration. So there's lots of ways to convert the acceptor intensity to volume, or sorry, to concentration. And so to do that, you have to use some proxy for the volume of the cell. And that's, of course, important because not all cells are identical, right? And so here we just have a schematic, paying attention now to these two bottom cells in the schematic. So you can imagine two cells with the exact same concentration of protein, but they have different volumes, different sizes. And so that's going to cause noise on your x-axis, or larger cells are going to be right shifted relative to smaller cells, even though the concentration, the thing that really proteins care about, can be the same. And so you can, if you correct for that, if you divide the expression level by the concentration or by the volume, then you get concentration, and those two cells will collapse where they belong, right? So there's, as I said, there's lots of ways to do this. Here I'm just showing you the relationship between three different proxies. So as I mentioned, imaging flow cytometry, that was how DAMFRET was initially developed. And basically what you can do is you can have the image of the cell, you can directly extract from that the radius, and from that you can calculate the volume, assuming it's a sphere. So you can, get the direct volume of the cell. And you can correlate that with side scatter. And that turns out to be, side scatter turns out to be a great proxy for volume. In this case, you can see this linear correlation. You can also use protein expression, global protein expression, or a proxy for global protein expression as a proxy for volume. So of course, as the volume of a cell increases, that's typically because it's producing more protein. And so if you just look at the expression level of any housekeeping gene, in this case, we use a PGK1 promoter, a housekeeping promoter, to drive expression of an orthogonal fluorescent protein, a far red fluorescent protein, BDFP. And if we simply use BDFP fluorescence as our proxy for volume, you see again a linear correlation with both the imaging derived cell volume as well as the side scatter. So you can do this, and that's great, because now that frees you from constraints on on biological system. Are there, for example, if your cells have a wide variety of shapes, right? Maybe they're red blood cells, I don't know, right? They're not going to be spherical, so you can't easily calculate volume from them. And side scatter depends on the nature of the cell, right? If it's neutrophils, for example, it's not going to have the same side scatter or the same relationship of side scatter to volume as other cells. So this is a way to get around that. Importantly, it also allows you to do this without an imaging cytometer, right? Most labs have access to a standard flow cytometer, not an imaging cytometer. And now you can do this very accurately without imaging cytometry. So that was an important update. How does that work in practice? So here we're looking at a couple of different phase separating proteins. This VPGVG is an elastin-like peptide repeat, and it will undergo condensation as a function of the number of repeats. So here we're looking at 45, and you can see if we're just looking at the raw DAMFRET plot, so acceptor versus AmFRET, or AmFRET versus acceptor rather, you see this transition happen. So the protein accumulates in cells as a monomer, and then it will begin to form a condensate. Likewise, this card9 card will initially accumulate as a monomer and then transition, in this case, to an ordered polymer. And you can see there's this relatively tight transition, but there's still some noise about that transition. And so now if you correct either of these plots by volume using either forward scatter, side scatter, or BDFP, you can see that the width of this transition becomes much narrower. So now, especially focusing here on side scatter, we can collapse this relatively broad transition to a much tighter transition. You can see this really nice kink in this flow plot, and that kink precisely tells you where the phase boundary is, or the solubility limit of that polymer is. Right? And so you can, in principle, convert this to actual units of concentration, like micromolar. That's a little more complicated, and we can tell you more about that. Or you can do is what we typically do, is just do it comparatively. And so that's just quantified on this axis over here. And this on the left just shows you the expected relationship of the repeat number of VPGVG to the phase boundary, right? So as we increase the length or the number of repeats, our phase boundary shifts left. So that means the condensate becomes more and more stable. And again, so this data actually tells you that, sort of to our surprise, that side scatter is actually a great proxy for yeast cell volume, and specifically the protein accessible volume of the cell, right? So not the total volume, including the organelles, but really what in the cell can your protein of interest actually access. So this is the volume that truly matters for the protein. And so now some of the fun things that we've done since developing these tools is we can now distinguish kinetic from thermodynamic barriers. We can detect sub-resolution condensates, identify sequence determinants, biological determinants, and map toxicity to specific phases. And I want to go through a few examples showing you how that works, or showing you that works. So here is a protein that is very well studied. So this is the prion-like C-terminal domain of human protein TDP43. And TDP43 is very heavily studied, in part because it's very closely associated with multiple neurodegenerative diseases of aging. For example, TDP-43 undergoes some aberrant phase transition in 97% of ALS cases. That is to say, in the neurons of these patients, there's aggregates of the protein. And so if you look at this protein in yeast cells, in this case, you can see a very complex DAMFRET plot. So the protein initially accumulates as monomer. There's now a transition. So we see an apparent phase boundary indicated by this dot, this dashed line, where it now accumulates in some higher order species. And then there's also a second transition to an even denser species, so even higher FRET. And as opposed to the first transition, which really was a function of concentration, the second transition is relatively independent of concentration. And so that suggests that second transition is really the amyloid state of the protein. We can confirm that simply by eliminating a factor in yeast cells that predisposes prion-like proteins to form amyloid. That is a 10, we convert the cells to 10-, so they now lack that cross-seeding amyloid. And now you can see that in those cells, you only have two states of the protein. The amyloid state is now gone. That means that these condensates can no longer nucleate. There's no longer a template to access that amyloid state. So that tells us that we can use this and the relative dependence of each transition on concentration to distinguish kinetic from thermodynamic barriers. We can also, of course, probe sequence very easily. Again, this is a single genetic construct. So in this case, we looked in the literature to find mutations that were known in vitro to either lower the phase boundary or increase the phase boundary, and we see exactly the same effect. So here we stabilize an alpha helix motif in the protein that drives phase separation. And when we do that, we lower the phase boundary. So the red line shifts left relative to the wild type line, the gray dot dash line. Conversely, if we destabilize that helix, we can reduce phase separation. And indeed, we greatly shift right that key position. Yeah. So we can, use this to identify sequence determinants. Now, interestingly, if this is imaging flow cytometry, we can derive this directly from the plots. We can also do this offline by correlating fluorescence microscopy with the DAMFRET plots to look at where the protein is in the cell in each of these phases. And so in the low arm of this plot, it's monomer, so it's this diffuse population. In this upper arm, surprisingly, there's actually no detectable condensates, even though we know that there's a phase boundary. And the phase boundary can be modulated by factors that we know control phase separation in vitro. And so that tells us that this protein is actually undergoing phase separation, but the condensates are not achieving a large enough size to be observable by microscopy. And this is a very important point, right? Because we often use puncta as to tell us if the protein is actually phase separating. And we're finding in many cases, this is one of them, that that's not actually always accurate, right? So there's a lot of condensation happening that you can't see by microscopy. And then if we look at this high FRET state, the super high FRET state, we can see the aggregates you would expect for amyloid. And so that tells us that you can use DAMFRET to detect sub-resolution phase behavior condensates. You can also detect biological factors. So in this case, we knocked out an HSD70 in yeast, and when we do that, we shift the phase boundary right, and we simultaneously increase or accelerate the nucleation of condensates to amyloid. And so that's as expected. And so we can use this then to identify biological determinants of phase behavior. We can do more complicated things. So of course, again, we're looking at the relationship of aggregation to concentration, and that relationship tells you a lot. So here I'm showing you from a recent paper where we used DAMFRET to identify the nucleating conformation of polyQ amyloid. And that involved generating lots of different sequence permutations of polyQ, and those aren't important in this case. What I want you to focus on are these curves in general, where we're looking at the fraction of cells in the high FRET state as a function of concentration. And what you can see is that for most of these proteins, there's multiple parts of the curve. Whoops. So in this case for polyQ, initially your AMFRET, your amyloid forming population increases dramatically up until a point, and there's a plateau or a shoulder where you have a regime where amyloid formation is no longer responding to concentration. That is to say that high concentrations are actually blocking amyloid formation. And then there's a second regime here where you now have hypothetically a different kind of aggregated state that's now increasing. And with certain mutations, you can actually block that second state from forming. So you actually have a plateau, in this case for this red line, where there's really, you're only able to form amyloid at low concentrations of the protein in cells. This is quite surprising, but ultimately what that tells us is that there are, for polyQ, it frequently goes into an off-pathway multimeric state that's not conducive to amyloid formation. Again, I mentioned this is all done on a single genetic construct, and what I've shown you so far are just rational mutations we made to different proteins. But moving forward, we're actually doing this, and we're not trying to be so clever. We're really just trying to do this in high throughput with random mutagenesis, taking advantage of tools like AlphaFold to then extract structural information from the pool of mutants. And so here's an example where we've taken an amyloid forming protein, a functional amyloid, mutagenized it, random error-prone PCR to mutagenize the protein. We then fax the high and low FRET populations at the same concentration sequence to determine for every mutation, what is the distribution between high and low at the same concentration? And that tells us, in this case, exactly, it gives us this hotspot, as you can see by this sort of blue column, where clearly that part of the protein is critical for amyloid formation. And then we can actually use, we can show with an AlphaFold predicted structure that corresponds to the core of the amyloid multimer. And now I think this is probably my last example, but so of course we can do this in high throughput for biological determinants as well as sequence determinants. And so here I'm showing you an example of some of the assays we do in the lab where we're taking a single protein, we're expressing it in thousands of different cell lines. In this case, we're doing it in the yeast deletion collection. This is robotically done. It's put onto a ZE5 cytometer where the collection rate is absolutely astonishing. This is actually in real time. You can see it's going from one well to another well to another well. So it takes about seconds to collect hundreds of thousands of cells. And so this is the output of one of those screens. So here looking at what happens for each of the 4,500 gene deletions to the nucleation barrier of that amyloid. And you can see, for example, here, gene deletions that dramatically increase amyloid formation and gene deletions that dramatically decrease amyloid formation. Okay. Oh, sorry. One more example. Now, importantly, what does any of this matter, and how does this matter for proteopathy? So we noticed in looking at these different volume proxies that when we're using global protein expression or proxy for global protein expression as an indicator of toxicity, we actually saw some populations on the DANFRET plot, most of them just sort of collapse. So we're getting rid of the noise associated with different volumes. But we saw that some of them actually systematically shift. They shift right. And when we thought about it, that actually, well, of course, that indicates that because our proxy for protein expression is in the denominator here, that says that in those cells, protein expression has been turned down, right? So it's inhibited. And that's actually one of the first and global things that happens in a stress response, right? Is when there's some proteotoxicity or otherwise other stress, protein translation is dampened. And we're actually now reporting on that. And that gives us a way to see which states or which populations on our DAMFRET plot are causing toxicity. And so now I'm showing you this here for a protein that we've characterized quite well. This is sub-35 prion domain. It's an amyloid forming sequence. We used some scrambles of the protein, just sequence permutations that were developed by Eric Ross's lab a few years ago. And when we express those in cells, so this particular version, number 21, is not associated with toxicity. So the cells grow fine when the protein is turned on, but this number 24 is highly toxic. And we can show how those, what do the phase behaviors look like of those proteins on a DAMFRET plot. And so the one that's not toxic actually has a monomer population and an amyloid population, but that's it. The one that is toxic has a monomer population, a couple of amyloid populations, and then some intermediate metastable population. And we can show that it's that metastable, that intermediate population, that is specifically causing toxicity. So here, this was the raw acceptor in DAMFRET plot. Now, if we divide the acceptor by BDFP and quantify the shift in the population between those two plots, you can see that the wild type protein, there's very little shift. So neither the monomer nor amyloid is really inhibiting cell growth or volume expansion rather. But in this case, for this number 24 mutant, specifically this population here is dramatically right shifting, indicating that in those cells, our housekeeping gene expression has been turned off or turned down, right? So indicating toxicity. So to summarize, we use DAMFRET to dissect protein phase behavior in living cells. We can use it for all these applications I just mentioned. In addition, something that I didn't mention is that you can do this now for the exact same protein with exactly the same assay across wildly different biology. So you can compare the same protein in yeast, in human cells, in neuronal cell lines. We even are now developing it for bacteria, and we're also developing a purely in vitro analog of DAMFRET. So this would allow you then to determine really precisely what is both the sequence and biological determinants of complex phase behavior. So I want to thank the team that made all this possible in the lab, beginning with Tariq, and then subsequently Tej, Shriram, Jinzang, Alejandro Taylor in Brooklyn. Some of them are still with us, others have moved on. Again, I want to point out Shriram, who's here with me today, and then as well as other members of the broader Stowers community, that helped us with this, as well as my funding. So, thank you all for listening. All right. So looks like we have some questions that have come in. I will welcome anybody, if that's possible. I don't know, Ronald, if anybody has any questions, they can just unmute themselves and ask directly. Otherwise, we'll just go with what's shown here. All right, Shriram, is there any outstanding questions? No, looks like.
Yeah, so Mohammed Mozamel Haik, so he has some questions on querying toxic phases of proteins, and also on dissecting the amyloid nucleus, nucleating structure of an amyloid.
Okay. Mohammed, if you are able to, feel free to just ask directly. Otherwise, I can read this and try to answer as best I can. Okay, so he says, okay, I need to know the structure of the transition state of the protein when it converts into the insoluble form, amyloid, basically the reason behind the conformational change of protein. When the protein is in the soluble form and in the insoluble form, what will be the transition structure of the protein? So the way to do that, it's quite challenging, but you can see, Shriram, did you provide the polycube paper? Is that the—
Yes, I did.
Referenced. Okay. So basically what you can do is the way to do that, it's challenging, but you can deduce the transition state, at least in principle, and that's what we argue in this paper, by exploring a large number of mutations that influence the DAMFRET plot. And so, because you're looking at the difference between high and low FRET and not necessarily the actual final structure, all your mutations are really probing are the residues that are absolutely required, right? to form that structure. So what is the core? And the core, but the kinetically most important things that have to happen to form that amyloid. And so when you do that, you can use all of those different mutations that block that transition, or that kinetically block that transition, to use those as constraints in different models. You can use that to come up with models of what that transition structure would look like. And then, and if you do that enough, as we did, I think exhaustively, we looked at over 300 rationally designed mutations of polyQ, you can eventually narrow it down to a relatively singular structure. Right?
So specifically, I think he's trying to get a feel for why polyQ amyloid is not purely concentration dependent, and also what is the reason for the conformational change from a disordered monomer to an amyloid? Okay.
So polyQ is a great example to think about this question. So obviously, as you know, amyloid is a multimer, so you would naively think that it's going to be concentration dependent. Oh, actually, I can see him raising his hand now. Let me see if I can unmute. Is that possible? Do I have that power?
Yeah, he should be unmuted.
Are you unmuted? I see his hand raised.
So may I ask you the question directly?
Yeah, please.
Okay, so your paper is based on the insoluble form of protein into the soluble form of protein converted into the insoluble form. Yes. So
Yes.
what is the structure of the soluble form in the protein? So what type of structure present in there, and when it will be converted into the insoluble form means amyloid is insoluble, and what type of structure present in the amyloid? So it is the alpha structure fully, or alpha beta both, or only the beta structure in there? And when it will be converted from insoluble form to soluble form, there is a transition will happen between the two form. Like so what type of transition will be happening there, and the region behind the transition? So you have shown in your paper, so it is not dependent on the concentration, so it depends on something others, like temperature or something, or pH. I don't think so. pH, temperature, maybe temperature. So when temperature will change, like physiological temperature or something will change, so what will happen of the structure from alpha structure into the beta? And the toxicity, my second question is toxicity.
Let's focus on the first question. Settle down. All right. All right. So the first question. What's happening, right? So if it's not concentration dependent, what is it? Keep in mind, right, that there's thermal fluctuations, right? The Boltzmann constant, describing the distribution of these fluctuations as a function of temperature. So you're right, temperature will influence it. We didn't probe temperature in this case. We're doing everything at physiological temperature. But at physiological temperature, proteins are constantly fluctuating, right? There's constantly some motion happening, and there's some distribution, right, of the magnitude, the distance, the magnitude of the fluctuation relative to the quote unquote native structure. So that describes the ensemble.
Yes.
Somewhere out on that distribution, we're showing that there is some extremely rare conformation of the monomer, right? And that is the rate limiting thing that then templates amyloid. That is the nucleus. So it's concentration independent. That means it's in a monomer. And it happens because it's just a really, really rare state in that ensemble. Very, very low concentration state in that ensemble. The concentration is so low, right, that there's less than 1 incidence of that species in a cell, right? And so that's why there's this discontinuity that's relatively independent of concentration. And so what we can do is, what is the monomer structure? We don't actually know, right? We know from other work. So lots of people have simulated this protein and done CD and all this stuff. And the monomer structure is disordered. It's a collapsed, extremely viscous, but extremely disordered species. There's no, for a long piece of polyQ, there's no helicity, even though a shorter stretch of polyQ does form a helix. And AlphaFold will tell you with 90+% confidence that it is, or PLDVT, that it is a helix. But for long polyQ, that's not true. It collapses on itself. It's self-solvating. It's a disordered species, but it's so viscous, right, that it just takes a really long time to finally find that beta, that intramolecular multiple beta strand conformation that is in the right orientation to form an amyloid core. And what we showed in this paper is that based on this pattern of mutations that inhibited the transition, we found there was this, and you may be familiar with this from the paper, but we found that there was this this parity dependence. So when we introduced mutations only at odd positioned intervals, we were able to form amyloid fine. When we introduced it at even intervals, we blocked amyloid formation, provided we did that at a high enough density. And so that tells us that as long as we have, it tells us a feature that's only explained by beta conformation, right? In a beta strand.
Beta, yes, beta conformation. Yes,
right.
yes, I agree. But when it will be mutated, so the region, so what is the region behind the protein structure will be mutated? So what is the region? So it does not, you have shown it does not depend on concentration. So it depends mainly something other. So what is the region for mutation? So mutation means structural change. Yes, structural change means
Yes.
it has a hydrogen bond. So hydrogen bond mainly
Yes,
makes
you
the
can think
secondary
about it that
structure.
way. Yep.
So hydrogen bond will break. So hydrogen bond will break, then it will be like a rod-like shape or something others, but it will not be stay like as a rod-like shape. So it will change something other. So maybe it can be changed into the beta structure. I don't know, because this is my question in here. So when it will be mutated, what factors work behind this? Which factor mainly? So what is the
Mutated in the
region
conformational
actually?
sense.
Yes, conformational change. So it will conformational change means different structure,
So
different
the factors,
conformation will have
are
spatial arrangement, side.
yeah. So okay. So right. So there's multiple ways to answer this question, right? And so there's going to be circular factors. There's any number of those things, right? Because homogeneous nucleation never happens in biology, right? I would argue never happens in biology, because there's always going to be some surface somewhere, something that has some affinity for the protein that's going to influence that's going to make it depart from the maximum nucleation barrier you would imagine if it was totally in a vacuum. However, if it is totally in a vacuum, as you can simulate, right? The only way to do that is really by simulations. And the simulations show that the protein at normal temperature, right? Temperature is really describing the fluctuations in the molecules, right? Of course. So it's not a single species. It's an ensemble. It's always moving. There's no other factors required but temperature. Temperature alone is responsible ultimately for nucleation, right?
Exactly.
That's providing the motions in the different bonds and the bond breaking, et cetera. That's always happening no matter what, unless you're at absolute zero. And eventually, right, there's a conformation. There's some combination of those tiny fluctuations that describes the nucleating structure.
Yeah. Okay. So I understand. So toxicity. So I'll ask.
Okay.
The second question.
Yeah. Uh-oh, you're breaking up.
Toxicity.
Okay, go ahead.
Is at the mono.
Toxicity with respect to polyQ. We don't know.
So which state is toxic? I don't think so. It will be toxic. So monomer is insoluble. I think so. For example, prion. So you have shown the prion. Prion is like a smallest size of the protein. What do you think? It's the smallest size of the protein. And so what is the structure in there? What form of structure in the prion?
In the prion?
Yeah.
So you mean the prion protein, or any prion generically?
Generally means toxicity. So I need to know about the toxicity. So I am very much curious about the toxicity.
Yeah, me too. Everybody is, right? So this is like the big things. Like, can we finally figure out what the quote unquote toxic species is? I share your enthusiasm. The short answer is we don't know, right? I mean, this is one step to getting us closer, but we don't know, right? There's lots of arguments to be made for lots of different species. It may be a whole combination of species. I think it's probably that. I mean, I think the general themes, right? And I'm sure you know this, but just for to put it out there, the general paradigm is that the native monomer, of course, is not toxic. The amyloid is relatively non-toxic. It's more toxic, tends to be more toxic than the monomer, of course. but because there's so low surface area, right? So it's in some aberrant form, but oftentimes because it's deposited, right? It's not interacting with a lot in the cell. And so that tends to be, for many proteins, relatively non-toxic, but it's these metastable species, right? And you're asking, what are those species? Is it beta sheet? Is it alpha helix? It seems like it tends to involve beta sheet, but not exactly the same thing that's happening in amyloid. It could be, I think historically we've thought about it as these are things that are kinetically en route to amyloid. I think that's a bit not quite accurate, because in many of these diseases, it's the nucleation event that's rate limiting. So you don't have these things happening. You wouldn't have amyloid association to the extent that the amyloid process is responsible, right? Unless the metastable species are downstream somehow, right, of the amyloid. And really influential work from multiple labs, but probably most convincingly from that of Thomas Knowles, is that in the case of A beta, the toxic oligomers seem to be catalyzed on the surface of the amyloid. So you have a rate limit. So amyloid nucleation is still hypothetically rate limiting. The amyloid grows. And as long as you're still at a high enough concentration, before it's totally reached equilibrium, right? You're still at high enough concentration so that monomers or oligomers will somehow interact with this lateral ordered surface. And that influences the conformations. That causes some conformational change in those oligomers. And those oligomers can diffuse away and wreak havoc on the cell. Eventually, they still wind up in the amyloid, right? Because the amyloid is the lowest energy state. But it's that intermediate, that region where this thing is undergoing exponential amplification and these things are diffusing away like crazy is presumably, according to them, what is the toxic, the toxic species in the case of A beta. In the case of polyQ, my suggestion, my speculating, and this is what we kind of led to in that paper that Shriram linked to you, is that the toxic state is post-nucleation, right? Again, so nucleation is happening in a monomer. And we showed that there is a phenomenon called self-poisoning, that where at high enough concentrations of monomer, the nucleating monomer cannot grow. So it's actually becomes kinetically arrested. There's sort of a traffic jam, if you think about it that way, happening during growth. And we think those are the species that are toxic, but we don't know, right? So we don't yet know. In principle, we could use the assay that we've talked about, and specifically this way to assess toxicity to try to get at that. We haven't done that yet.
Okay, so means conclusion is, so beta structure of the protein is mainly toxic. It can be either monomer or can be polymer. Yes.
Yeah.
Yes. And also the side chain. So if protein side chain is hydrophobic, it can also be the toxic. So if protein has a hydrophobic side chain, I think it can also be the toxic. So means hydrophobicity. So when alpha structure convert into the beta, so beta structure is hydrophobic. What do you think? So beta structure is almost hydrophobic. Alpha structure is not hydrophobic. And the short chain. So short chain and the beta structure and the side chain. So 3 points are mainly responsible for toxicity. Yes.
Oh, I think it's hard to generalize that much. Right, because right.
Yes. Okay.
So secondary structure. Anytime you have structure, right, that involves hydrogen bonding, you're becoming more hydrophobic. That's going to be true for alpha helices. That's going to be true for beta sheets, right? And then
yes. So, for example, when we look at the disease, this is called ferritin. So ferritin is a iron overloading protein. So iron overloaded protein in there. So this is also hydrophobic. So ferritin, we see their structure is only beta structure in there. And iron, when iron lose the 1 electron, it convert into the hydrophobic. So hydrophobic interaction and protein goes collapse. Like it can convert into like a mold, a small mold, like it cannot be water soluble. So this is why the ferritin is toxic. So thalassemia patient we have found there, and there the ferritin is high in there, iron overloaded, but there we find the iron is hydrophobic. So means if protein has a hydrophobic side change, or if protein lose the structure of alpha structure, loses. So it can be toxicity. So ferritin is highly toxic. So fluoride, for example, fluoride also shows the hydrophobicity. Fluoride. So fluoride ion, we have found the fluoride is hydrophobic, and fluoride is also, we see the toxic, particularly especially is highly toxic for protein. We have found like this. And then the so
I
many
guess let me try to summarize. I think there's, I mean, cells are complicated things, right? And there's a gajillion things that have to be working coordinatedly, you know, cooperatively to make life possible. And I think you can throw a wrench in any one of those things, right? And so I don't think there's lots of ways to kill a cell, right? And so there's, it may be different for each protein. In fact, genetic screens suggest that each of these amyloid preferring proteins may have different mechanisms of toxicity, right? There's probably some common themes, but the factors, the proteins that they are hypothetically interacting with, aberrantly, maybe because of higher avidity in the oligomeric or aggregated state, are going to be different depending on what the array of side chains in the aggregated species is. That's going to determine what they interact with in the cell and what's going to go wrong, right? And so I think it's really I don't think we're going to gain much by generalizing too much. Unfortunately, I don't think it's going to be that simple to determine what the toxicity is. Right?
Okay. Thank you.
Thank you for your questions. Yeah. Shriram, did you have something to contribute to that? Or?
No, I just wanted to alert that we have one more question. If we have just enough time to quickly answer that.
Okay, sure.
So Vicent, he or she asks, is there a way to capture an intermediate state with this technique to dissect the structure?
I think you could enrich for it. I mean, I think in principle it could help you. I think it would be very challenging. So you know, let's take the example of the SUP35 number 24 mutant, right? There's a relatively well-defined population of cells that had some metastable species. So you could imagine enriching, right? You can definitely use FACS to enrich that population of cells. And then what you do with them after that becomes the challenge, right? So you could, obviously, you could try to lyse the cells, extract and purify that protein, and then do cryo-EM. But I think in the process of doing all of that, you're going to end up converting, right? Because again, once nucleation happens somewhere in the system, it propagates. And that's really, I mean, so DANFRET really allows you, because it's compartmentalizing the global volume into tiny little volumes. Nucleation and these metastable species are relatively stable kinetically. So once you lyse the cell, and now all the protein that was previously confined to individual cells is now exposed to every other molecule. In principle, once nucleation happens, it's gonna, it has the, you know, unless your state is really stable, you know, it's all going to end up in the amyloid, and you're going to lose that species. But I don't know. I mean, you know, there may be other. Certainly, there are ways to try to get there, but I think it's going to be challenging. You could also imagine just doing it. So it's in principle, and people have done this. You can actually just FACS aggregates themselves. So if you have an in vitro system where you think you're achieving some appreciable population of the metastable species of interest, then you could, in principle, just FACS it directly. But man, that would be a lot of FACS time to accumulate enough particles to then do cryo-EM or something with them.
All right. So another way to quickly interpret this question is, what if the intermediate state is not, is super transient, like the nucleating structure of polyglutamine? So in that case, we can only deduce the structure by putting a lot of, structural constraints, by imposing a lot of mutations, analyzing all that data, and we can't directly capture the structure, and that's what we have aimed to do.
Thanks so much for that, Shriram. Yeah, that's the main point. Actually, is that back to the original slide where I was talking about all the challenges in doing this. It's that there isn't a way to do it, right? Until, arguably, at least until this, and just echoing Shriram's point, maybe elaborating a little bit, is we can't directly, even with this approach, we cannot directly understand what those metastable species are, but our approach can specifically tell us about that highest energy state, right? Which I think is the most interesting in terms of, because it's obviously the most important in terms of kinetics of whatever the final state is. And so again, if you did, and this is something that my lab is really excited about, in principle, if you mutagenized your sequence deeply enough, and again, you can do that because it's a single genetic construct, and then you sort all the species that have the right characteristics, right, at the right concentration, and you look at that distribution, and you sequence those, then you have suddenly thousands of sequences that specifically form the structure of interest. Hypothetically, depends on how confident you are that they are forming that structure, and a whole bunch of sequences that don't. And with that sorted list of sequences that can form the structure, in theory, right, if it's deeply enough, you will be able to extract couplings between positions that give you structural constraints, right? Statistical couplings between positions that give you structural constraints on what the actual structure is. And this is exactly what AlphaFold does, right? But AlphaFold does it with natural multi-sequence alignments. What our technology allows you to do, in principle, is to do exactly that same thing and use AlphaFold, but replace the multi-sequence alignment from the natural sequences with your artificially selected pool and obtain structure. So that's what excites me about this technology ultimately. Okay, Shriram, did you have something else to?
Yeah, before we go ahead, Ronald, do we have time to answer one more question quickly?
Yeah, sure.
Okay, so Constanza asks, I was wondering if you used other approaches to confirm that there are condensates even when you do not detect puncta formation, as in the case of maybe say TDP43, I think.
So in the case of TDP43, we tried multiple other things. Well, we didn't try that many things. We tried super resolution imaging. So we tried STED microscopy, and we could not see anything. And I think the reason for that is that the concentration, the phase boundary is so high that you just can't distinguish. I think if it was low enough concentrations of the protein, STED would allow us to see puncta, and you could potentially look at the brightness of those and try to get it. You would be able to distinguish monomer from something else. But at these concentrations, it's, double-digit micromolar concentrations where this is happening in the cells. You can't see anything. It's still diffuse. It's bright, but it's still diffuse. And so the short answer in the case of TDP43 is we don't have any other evidence. But FRET is FRET, right? I mean, FRET is not lying to us. And we know that the molecules are interacting. We know that that's concentration dependent, and it has a tight concentration dependence, indicating it's a very cooperative species that's, large, then relatively large. And again, it responds we can extract structural information on what is holding that species together, right, in a rational way based on structures that have come before us. So no, indirectly, we are confident about it, but we haven't found any other approach, or we haven't tried anything else beyond imaging to try to confirm that those species are there. Good question.
Okay, so it looks like that's it as far as questions. So just like to thank Randal again for an excellent presentation here. And as I said before, we did record the webinar, so we will send the recording out by email for your records, and you're welcome. If you have any other questions at that time, for Randal or for JoVE.
Or Shriram. Yeah, I want to thank Shriram as well. For
Shriram as well. You can feel free to ask them then. So yeah, thanks again everyone for attending, and enjoy the rest of your day.
Thank you, Ronald.
Thank you.
Take care. Thanks again.
And anybody who's listening and has questions, you're free to email me. I will happily respond. So thanks so much.
All right, thanks again. Bye-bye.
Okay, bye-bye.
Bye.